Today, we're doing something a little different and honestly, something really exciting. Our mission is to reverse engineer exactly how top-tier science is built. We are going to deconstruct 3 massive neuroscience papers from Nature just to see how we can turn incredibly dense literature into a blueprint for a brand new major discovery.
Okay, let's dive right into this roadmap. We'll kick things off with the scientific deduction method. Then we'll map out 3 groundbreaking brain studies, hunt down their blind spots, synthesize a massive nature-level idea, and finally, figure out exactly how to prove it.
All right, part 1, the scientific deduction method, or basically how to build a masterpiece.
We're going to approach these papers like a detective at a crime scene, looking for the clues that everyone else just walked right past. Step 1 is rapid structural scanning, getting the lay of the land. Step 2, ask critical questions to push way past the raw data. Step 3, we do a horizontal comparison to spot the gaps between the papers. And step 4, the fun part, we generate an original idea to fill those exact gaps.
So let's move to part 2, 3 groundbreaking brain maps. It's time to step into the evidence room.
All right, let's look at paper number 1, and this is driven by the International Brain Laboratory, or the IBL. Guys, this is a staggering paper. It gives us the first ever brain-wide map of neural activity during complex behavior in mice. And the sheer scale here is just wild. We are talking 279 distinct brain areas and over 620,000 single neurons. By using this standardized open access data set, it completely shatters that old assumption that decision-making just happens in one localized little hub. Instead, it turns out, decision signals are distributed literally across the entire brain.
But here's the critical question you've got to ask yourself. Does this massive, beautiful map actually explain the cause of a decision? And the answer is absolutely not. It's just a descriptive mapping exercise. It shows us what lights up, sure, but it is completely correlational. It's not causal. There's literally no explanation of the actual mechanism behind how these different areas collaborate to process specific parts of a decision. And honestly, that is a massive structural blind spot.
Okay, moving on to our second piece of evidence. This is another incredible Nature paper, and this one focuses on prior information. Basically, that's the subjective historical probabilities our brains use to make choices. Using some incredibly advanced tools like neuropixels and calcium imaging, which, by the way, literally let us track electrical spikes and chemical flashes in live neurons, the researchers proved that these priors aren't just stored in a single filing cabinet. They're processed everywhere, in parallel, across multiple processing layers. Now, what's really interesting about this is the huge gap it reveals. Because, okay, we now know where these subjective priors are located, which is essentially everywhere, but we have absolutely zero idea how they work mechanically. I mean, is it a dynamic weight adjustment? Is it some delayed feedback loop? We don't know. The paper doesn't know. There's no functional comparison between different encoding types, and absolutely no proof that this even generalizes across different types of tasks. Which brings us to paper 3, a highly relevant study from Nature Neuroscience. This team hit on a major headache in modern research. Think about it. When an animal makes a decision, it usually moves, right? So are the neurons firing because the animal is actually thinking, or just because it's moving its body? To figure this out, they combined behavioral video with single neuron spike data, and they used machine learning to try and untangle the movement signals from the cognitive signals. And guess what? They found that during these behavioral tasks, a massive majority of neural signals are actually just related to movement. But while this paper is fantastic at waving a giant red flag and identifying that interference between movement and cognition, it kind of stops short of the finish line. It doesn't actually explain the fundamental computational difference in how true cognitive signals are computed versus how motor signals are just executed. So that leads us right into part 3, finding the blind spots through a horizontal comparison. When we take these 3 papers and lay them out side by side, we expose what I like to call the illusion of knowledge. Look at this. The brain-wide map shows us distributed decisions, but totally ignores movement. The prior encoding paper shows us widespread subjective data, but gives us zero mechanics. And the movement model paper shows us that movement pollutes our cognitive data, but doesn't explain the deep functional differences. So, putting it all together, none of these top-tier papers successfully isolate true cognitive computing. So the crucial point here is this conceptual fault line. Distributed encoding is absolutely not the same thing as distributed mechanisms. Just because the whole brain lights up like a Christmas tree when a mouse makes a decision, that does not mean every part of the brain is equally participating in the actual cognitive math of that decision. We're literally confusing the map for the territory. All right, part 4, a nature level idea. This is our aha moment. By synthesizing all those blind spots, here is the proposal that emerges. Ready? Develop a causal, task-independent framework that disentangles distributed neural representations into computational roles, sensory, cognitive, and motor, across brain regions using joint modeling of large-scale electrophysiology and behavioral video data, and validate causal contributions via perturbation experiments. Boom. That right there directly addresses every single blind spot we just uncovered. And why is this approach so incredibly compelling? Well, first off, we aren't reinventing the wheel here. We're building on the massive open data sets the IBL already collected. Second, we're finally shifting the field away from just descriptive, hey, look what lit up mapping, toward actual causal mechanisms. Third, we are integrating cross-modal data, smashing together electrophysiology, video, and machine learning. And finally, we're adding that missing engine of causality through perturbation techniques like optogenetics, basically using light to directly control specific neurons. Which brings us to part 5, proving the concept. Let's talk execution. And this brilliantly illustrates how a lab would actually pull this off, step by step. First, you dive into that open IBL dataset. Second, you run machine learning encoder-decoder models, which is essentially translating complex neural noise into distinct categories to digitally separate out sensory, cognitive, and motor signals. Third, you take those digital predictions out into the real world. Use optogenetics to temporarily turn off specific cognitive nodes in a live brain just to see if the behavior actually changes. And finally, you test this across multiple tasks to prove your findings are a universal rule, not just a one-off fluke. By combining machine learning with physical brain perturbation, we completely bridge that gap between correlation and causation. It totally reshapes our understanding. So I'm going to leave you with this provocative thought. If every single part of the brain lights up when we take an action, which part is actually doing the thinking? And more importantly, if you were the scientist walking into the lab tomorrow, exactly how would you prove it?